Today we are going to discuss the CRD, which is completely randomized design. So this is one of the experimental designs, and in the previous lecture we studied that what is meant by the experimental designs, what is meant by the designing and planning of the experiments, and we also discussed that what are different components of the experimental design and what are the principles of experimentation. And there are three types of the experimental designs that are used in field research, and CRD, which is completely randomized design, is the first type of these experimental designs. The other two designs are RCBD, which is randomized complete block design, and Latin square design. So CRD, as its name indicates, complete randomized design, is the design in which treatments are assigned completely at random. And remember what is meant by treatments. Treatments are the categories of the independent variable that are applied on the experimental units to see the response of the dependent variable from the experimental units. So in CRD, the treatments are assigned completely at random at the experimental units, which means that each experimental unit has same chance of receiving any treatment. So if you have 100 experimental units, then each of these experimental units should have the same chance of receiving any of the treatments. If the number of treatments are, let's say, 5, then each of the experimental unit has the same chance of receiving any of the treatment. But remember one thing, that one experimental unit is going to receive only one treatment, right? But what treatment is going to be assigned to that experimental unit, it is decided completely at random. So when we when we apply these treatments to the experimental units, and then we record the response variable, and the response variable is our dependent variable. So any difference in the measurement of the response variable among experimental units that receive the same treatment is considered experimental error. Now, remember that what is the purpose of doing experiment? The purpose of doing experiment is to see the differences in response of experimental units to different treatments, right? So it supposes that the treatments that are, the experimental units that are receiving the same treatment, they should have the same response, right? But when we measure the response, so usually what we observe is that there is a variation in the response among experimental units that had received the similar treatment. So what is this variation coming from? This variation is coming from the extraneous factors which cannot be controlled, right? And this is known as experimental error. So CRD is appropriate for homogeneous experimental units like the lab experiments, in which the environmental conditions are easily controlled as compared to field, right? So because in CRD, the treatments are assigned completely at random, it means we are supposing that the experimental units are being kept under similar conditions, right? The environmental conditions in which the experimental units are raised, those environmental conditions are similar, they are homogeneous. So there is very little or minimum effect of the extraneous factors to cause experimental error. And the major source of difference left would be the difference because of the different treatments. So the CRDs, they are very simple and basic type of the experimental design, but they are appropriate under the homogeneous environmental conditions, which are usually applicable only in the lab experiments. So in CRD, we are going to assign treatments completely at random to the experimental units. So let's see a situation here. We have 30 parts, which means the number of experimental units are 30. And the number of treatments are A to C. So here we can see that that another requirement or another principle of the experimental designs is being fulfilled, which is replication. So replication means that the different experimental units, they are receiving similar treatment. And here we can see that we have 3 treatments and the number of experimental units are 30, so there are going to be enough replicates of the same treatment in this experimental design. And let's talk about the randomization, and the randomization here is completely fulfilled. The treatments are assigned at random. So there are 30 parts and each part is going to receive a single type of treatment, and the number of treatments are 3. So if they are given in equal replicates, then there are going to be at least 10 replicates of each treatment. And these treatments are applied completely at random, as you can see in this sketch, that there are 30 parts, and you can see that there is no specific pattern in which treatments are applied onto the experimental units. Rather, they're applied completely at random. And it is done at completely at random because it is supposed that all of these 30 parts, all of these 30 experimental units, they are being kept under same environmental conditions to reduce the experimental error. And now the sources of variation in CRD. So CRD is the simplest type of the experimental design, and in the CRD, the experimental units, they are kept under similar environmental conditions. So therefore, the sources of variation in CRD are very few. In fact, these are only 2. So the sources of variation in CRD are treatments and experimental error. So treatments is that source of variation which is of our interest. So this is the source of variation of interest because we are going to do the experiment to see the response of experimental units to treatments. So therefore, this is the major source of variation, and this is the source of variation of interest. The other source of variation, which is not the source of variation of interest, this is introduced by the nuisance factors or the extraneous factors which are beyond the control of experimenter, and they result into experimental error. So the purpose of experimental design is that you must reduce or you must eliminate the sources of experimental error to maximum extent possible. So the effective source of variation in CRD are the treatments, and the other source of variation which is reduced to minimum, so that is why that is not an effective source of variation, that is considered the experimental error. So these are the two main sources of variation in CRD. So the total variability that we see in the response of experimental units is the sum of variability due to treatments and variability due to experimental error, right? So the total variation is equal to variability due to treatment and variability due to experimental error. And by simply stating, we can designate total by TO, treatment by small t, and experimental error by small e. So this is the basic principle of CRD, and this is the basic principle that can be used for the statistical analysis of the data which is obtained by CRD. So let's do CRD with an example, and the example is effects of fertilizer dose on the fresh weight of plants. The experimental design is CRD, which is completely randomized design, and the independent variable is the fertilizer doses. Treatments are different doses of fertilizers. For example, we assign 3 types of the doses, A, B, and C. And the dependent variable, which is our response variable, is the fresh weight of plants, which we are going to measure, right? So our independent variable is that we are going to manipulate to define different treatments, and these treatments are to be applied on the experimental units, which are the plants, and the response variable is to be measured after the completion of experiment, and this is going to be the fresh weight of plants. So our experiment is to see the effects of fertilizer dose on the fresh weight of plants, and we are doing that under the experimental design of CRD, which is completely randomized design. So here we have the data, here we have the observations taken after the completion of experiment. So here you can see that there are 30 parts, there are 30 experimental units on which treatments were applied, and you can see the conditions of experimental designs being fulfilled. The first condition is the local control, and the local control was exercised by putting these parts under homogeneous environmental conditions to reduce the experimental error. And then the condition of replication, and you can see that there are enough number of replicates in this experiment. There are 3 treatments and there are 30 plants, and each treatment is applied 10 times. So there are 10 replicates of each treatment. And then the third condition is randomization, and this is complete randomization. There is no specific pattern in which the treatments are applied to specific plants, rather it is done totally at random. So this is the result that we have This is the data that is obtained. So in this data, you can see that the part number 1, or the experimental unit 1, was given the treatment A, and the response is 32 grams. The part number 2, or the experimental unit number 2, was given the treatment number B, and the response is 23 grams. The next experimental unit received treatment B, and the response is 48 grams. And the next one received the treatment B, and the response is 17 grams. And the next one received the treatment C, and the response is 42 grams. So here you can see that not only the response is different between the treatments, response is different within the treatments as well, right? So the reason for the differences in response within the treatments is because of the experimental errors, and the differences in the response between the treatments is because of the treatments applied. So here next we are going to analyze that whether the variation which is introduced by treatments is significant enough or no. So here we are going to arrange the data in columns according to the treatments. So we have 3 columns, A, B, and C, according to the treatments that were applied. And because the response variable is a measurement variable, so we can easily compare their mean values, and we can do that through one-way ANOVA. So why one-way ANOVA? Because there is only one type of the independent variable, and there is only one type of the measurement variable. Therefore, we can apply one-way ANOVA. And we are not applying 3 tests because there are 3 categories of the nominal or independent variable. So number of treatments are 3. So therefore, we are going to do it through one-way ANOVA. Now, after we are done with the experiment, we have obtained the data. So now it is time to do the statistical analysis, and the statistical analysis going to be done for this data is one-way ANOVA. So for that purpose, as a first step, we are going to make a table of descriptive statistics. And in this table, you can see that we have mentioned the number of observations, the mean values, variance, the summation x whole square, and the summation x square. So the number of observations in 3 treatments, the treatment A, B, and C, which are designated here as sample number 1, 2, and 3, are 10 in each. And the mean fresh weight of plants in treatment A is 36 grams, and the mean fresh weight of plants in treatment B, or sample number 2, is 31.6 grams, and the mean fresh weight of plants in treatment C is 32.4 grams. So you can see that their mean values are a little bit different. Now let's see their variances. So variance of the sample number 1 is 32.44 grams, and variance of treatment B, which is sample number 2, is 127.38 grams, and the variance of the third treatment is 53.6 grams. And then we have the summation x values, which are 360, 316, and 324 for the 3 treatments respectively. So here we have our descriptive statistics, and this is going to be used in the computation of ANOVA. The step number 2 is to test for the homogeneity of variance between the treatments, and this is to be done by the FMAX test. And what is FMAX test? It is the largest variance divided by smallest variance. So 127.38 divided by 32.44, which is the smallest variance, we are going to get the variance ratio, which is 3.93. And now we have to see that whether this variance ratio of 3.93 is significantly different from unity or no. And for that purpose, we have to consult the FMAX distribution table. And to get the critical value from the FMAX table, we need the information about number of samples or treatments, and V, which is the degree of freedom of every sample or smallest sample. So the number of samples or the number of treatments in this experiment are 3. So A is 3, and V is the degree of freedom of every sample or the smallest sample, because all of the treatments, all of the samples, they have the similar number of replicates or similar number of observations, which is 10. So we are going to take 10 to calculate the degree of freedom, and this is going to be 9, right? Now the calculated Fmax value is 3.93, and the tabled critical value is 5.34 at A3 and V9. So therefore we can see that our calculated value is less than the table value, Therefore, this difference in the variances is not significant, and variances are supposed to be homogeneous. Therefore, we proceed with ANOVA. So our step number 3 of ANOVA here is to calculate the correction term for ANOVA, and the correction term, or CT, is calculated by dividing the square of the sum of observations by the number of observations. So the sum of observations is 1000, and it is going to be divided by the number of observations, which is 30, and the answer is 33,333.33. And the step number 4 is to calculate the total sum of squares, and the total sum of squares is the summation x TO, which is the total squares, minus CT. So this is 35,364 minus 33,333.33, and the answer is 2030.67. So 2030.67 is the total sum of squares. Now we have to calculate the treatment sum of squares, which is between samples. So for that purpose, we are going to take the summation x1 whole square. It is to be divided by the number of observations in sample number 1 plus the same factors for the other two samples, and then their sum is going to be subtracted from correction term. So here we have these values, and we solve these values, and we see that the treatment sum of squares is 109.87, right? So now we have calculated two sum of squares, the total sum of squares and the treatment sum of squares. Now calculate the sum of squares for experimental error, which is the sum of squares within. So for that purpose, we are going to calculate the individual sum of squares, and the individual sum of squares are calculated by this formula, which is the summation x1 square minus summation x whole square divided by n1, which is the number of observations in sample number 1. So we calculate the individual sum of squares for each of the samples, which are 292 for sample number 1, 1146.4 for sample number 2, and 482.4 for sample number 3, which is our treatment C. And the individual SS is a sum of these, the experimental error sum of squares is the sum of individual sum of squares, and which is 1920.8. And in the step number 7, we are going to cross-check the values of these sum of squares. So because we have calculated all three sum of squares, total, treatment, and error, so sum of squares error is equal to the sum of squares total minus sum of squares treatment, and we can see that this is the same value that we obtained by adding the individual sum of squares. So we are done with the first part of ANOVA, which is calculation of the sum of squares. Then we have step number 8, in which we are going to calculate degrees of freedom for total, for treatment, and for experimental error. So the degree of freedom for total is NTO, which is the total number of observations, minus 1. So total number of observations are 30 minus 1 is 29, which is the degree of freedom for sum of squares total. Then the degree of freedom for treatments is A minus 1. A is the number of treatments, minus 1. So we have 3 minus 1 is equal to 2. And then we have the degree of freedom for experimental error sum of squares. So this is going to be the total number of observations minus A, which is 30 minus 3, which is equal to 27. And we can also calculate the degree of freedom for experimental error sum of squares by taking the individual degrees of freedom for all the three treatments and then taking their sum. So this is going to be 999, and their sum is 27. So here we have calculated the second component of the ANOVA, which is the degrees of freedom. Now we are going to step number 9 to calculate the MS, which is the variance. So MST, which is the variance due to treatments, variability treatments. So this is going to be SST divided by DFT. So sum of squares treatment divided by degree of freedom treatment. So we have 109.87 divided by 2. This is 54.935. And then we have to calculate the MS experimental error, which is the variance within. And for that purpose, we have the sum of squares of experimental error and this is to be divided by degree of freedom of the experimental error. So we have 1920.8 divided by 27, and the answer is 71.14. So here we have calculated the MS, or the variance, for treatments and for experimental error. So what is the next step? The next step is to compare these variances. And this is to be done by the F test. So in the F test, we are going to calculate the F value, which is the ratio of the variance treatments and variance experimental error. So MST, which is the variance treatments, is going to be divided by MSE, which is the variance due to experimental errors. So 54.935 is to be divided by 71.14, and the answer is 0.772. So this is the value of F, and here we can see that this value is less than 1 because the within variance, or the variance due to experimental errors, is greater than between samples or between treatments variance, which is the treatments variance of 54.935. So now let's see that are we going to have a significant result or no. And this can be checked by comparing the F value, which is 0.772, with the critical F value, and the critical F value is retrieved from the F distribution of one-tailed test, and for that purpose we need the 2 degrees of freedom, the degree of freedom for treatments, which is 2 in this case, and the degree of freedom for experimental error, which is 27 in this case. So the critical F value is 3.35 at probability of 0.05. So we can see that our calculated value is less than the table value, is less than the critical value, so therefore our result is non-significant. Now we put the results into the ANOVA summary table, and in the ANOVA summary table we have the first column, which mentions the sources of variation, and these are treatment and error, and their total is also mentioned. And then we have the sum of squares, which are 109.87 for treatment and 1920.8 for error, and their total is 2030.67. And then the next column we have degrees of freedom, which is 2 for treatment, 27 for error, and 29 for total. Then we have MS, which is our variance, and this is 54.935 for treatments and 71.14 for error. And then we have the F value, which is the ratio of the treatment variance divided by the error variance, and this is 0.772, and this is non-significant at probability of 0.05. So now it is time to state our results, and the result is there is no effect of different fertilizer doses on the fresh weight of plants. F at 2 and 27 degrees of freedom is 0.772, and the probability is greater than 0.05, right? So we have obtained a non-significant result in this case. So this is the example to explain that what is meant by CRD. So CRD is the completely randomized design, which is one of the experimental designs used in field research especially. But CRD is that design in which the treatments are assigned completely at random to the experimental units, which supposes that experimental units are to be kept under homogeneous environmental conditions to reduce the experimental error. So therefore this kind of the experiment is done under controlled conditions like in the lab. So in this example we saw that we have taken 30 experimental units, 30 plants, and these plants are given 3 types of the treatments, which are the different doses of fertilizer, and the experiment was done to find out the effects of these fertilizer doses on the growth of the plants by taking the response variable as fresh weight of plants. So the experiment was done, and the treatments were applied onto the experimental units in replicates of 10 each, and then the measurement variable was recorded, which was the response variable of fresh weight of plants. And then, because this was only one type of the independent variable for which we were testing, so therefore we applied one-way ANOVA. And this is the result of one-way ANOVA, and here we can see that there is no effect of the fertilizer dose on the fresh weight of plants. So this is how CRD is done.